Okay, great. I'm just going to back right out of here and I'm going to introduce, you met Andrew Weller previously. He is our board chair. And I'm just going to do a very quick introduction, if you don't mind, for Dr. Anil Kapoor. Some of you might be very familiar with Dr. Kapoor. He might be your doctor. And he is currently a professor of surgery, urology, and oncology at McMaster University and St. Joseph's Hospital in Hamilton, Ontario. He got his medical degree in Dalhousie, oh, I won't be able to say it, Dalhousie University down there in Halifax. So truly a good Canadian. And he is currently practicing and is one of the leading doctors in kidney cancer and innovative surgeon. And I hand it over to you to tackle the research element of it. There you go. And oh, Helen, I'm sorry, I should introduce Helen just in case. Helen is our Vice Kidney Cancer Canada. She's also a patient. And Helen, I'll let you explain what you did and when the time comes, okay? I'll leave it to you. Thank you.
So Kidney Cancer Canada strives to fund and promote leading-edge research. With this aim, we partner with the Kidney Cancer Research Network of Canada to request and evaluate proposals to award $50,000 seed funding grants for research within Canada. These awards are made possible through donations and specifically very generous grants from the William Soper Foundation. Portions of these donations are also matched by the Canadian Urological Association Scholarship Fund. I'll now turn it over to Dr. Anil Kapoor, one of our board members who you just had introduced, and he's also the chair of the Kidney Cancer Research Network of Canada, to discuss the impact of this research and introduce our award-winning researchers.
Thanks a lot, Andrew. It's great to be here. Welcome, everybody, on a bright, sunny Sunday, and it's a good forum so far. I enjoyed the wellness lecture. That was very informative and teach me how to breathe properly and meditate. Something I definitely need to get more practice at. So now we're going to go into the research portion of this, and Kidney Cancer Canada and KCRNC have had a great history of research. We had this organization called the Kidney Cancer Research Network of Canada, KCRNC, which is founded mainly by Dr. Michael Jewett and Dr. Simone Tanguay, in combination with Deb Maskins and Tony Clark about 12 years ago now. And with this network, we've been able to have an annual forum, research awards, and all sorts of initiatives to further kidney cancer research. And we've been the model for many other disease sites, such as bladder cancer, melanoma, thyroid cancer, and they're mimicking a lot of our research initiatives and infrastructure that we developed to further kidney cancer research in Canada. Along those lines, we've had a great collaboration with the William J. Soper Trust. The William J. Soper Trust was basically a organization that had a relationship with Tony Clark, Deb Maskins, and Michael Jewett, and over the last 5 to 6 years have donated prolifically to kidney cancer research. And so we do welcome them for their great support of kidney cancer research over the years. Last year, they donated $150,000 towards kidney cancer research, and I think they should be applauded for that contribution. Without this kind of funding, we're not going to be able to get a cure for kidney cancer. I think that's really what we want to do in the laboratory and clinical research. We really need to find a cure for kidney cancer, and we're making great progress. A lot of these immuno-oncology drugs we're going to talk about today have up to 10–15% complete response rate and other responses, all progress from research, not just in Canada, but around the world. We also have a collaboration with the CUA, the Canadian Urological Association. This is an amalgamation of all the medical oncologists, urologists, uro-oncologists, radiation oncologists across Canada in a global sense, not just in cancer, but all members. And the CUA has created what's called the CUASF, or the CUA Scientific Foundation. And they've also had an interest in funding kidney cancer, and they've matched the KCC grant. So KCC puts in some money through your donation. Many patients have donated. We appreciate all the donations, and with those donations, the CUASF matches dollar for dollar all those grants, all those donations, and also in combination with the William J. Soper Trust, we have these well-funded grants, and this year we funded $100,000 of grants into 2 main grants, but we had really 8 or 9 fantastic grants that were, or fantastic grant applications that were high quality research in kidney cancer. We were only able to fund 2 of them, and so we're going to talk to a few of those today that were awarded, but we want to fund all of them because all of them can further the progress in kidney cancer and help patients live longer and with better quality of life. So this year, that $100,000 was COASF money matched by Kidney Cancer Canada through your donations. And so again, a great appreciation to fund these young researchers that you see on the screen, Christine and Keith. Well, maybe Keith is not so young, but Kristen is definitely young. And so with that great, with all their great initiatives, we're going to really put a hammer in kidney cancer and hopefully towards a cure. So I'm going to announce some of the grants, and just for completeness, we're going to have Kristen and Keith talk today about Rola Celeb at the University of Toronto was awarded a grant looking at the validation of the biological classification system of papillary renal cell carcinoma to guide clinical management. And this is really to heart for Deb Maskins. Deb Maskins has talked over the years, and we all agree that we have a lot of research in clear cell carcinoma. So we have a lot of research in patients who have clear cell type of pathology of their kidney cancer. That's the most common, but then we have what's called non-clear cell histology. And these are cancers that are papillary type 1, papillary type 2, chromophobes or chromatoid. And there's not a lot of research happening in that arena for non-clear cell histology. So we awarded this grant to look at the biological classification system for papillary to help those patients with non-clear cell carcinoma. So I think that was a valuable grant, and we look forward to the research from University of Toronto and Rola Celeb. Mariam Soleimani, I hope I'm saying these names right, I believe I am. Mariam Soleimani is from the University of British Columbia, working with Christian Kolzenberger, and her award was for the exosomes of microRNA evaluation of biomarker development in patients with metastatic RCC treated with immunotherapy. Again, a long title, and I'll explain what that means. Basically, patients with metastatic kidney cancer, we now treat, and many people on the call may be already on nivolumab or pembrolizumab or any other such immuno-oncology drugs. And we're really looking for biomarkers to help guide our treatment. Not every patient responds, and we do know about 10% can have a complete response where their cancer is cured. Amazing, amazing developments, and certainly in sarcomatoid, you can have up to a 20 to 30% complete response rate. But those patients that don't get a complete response or don't respond, why is that? And can we tailor the therapy to those patients? And so Mariam Soleimani and Kristen Kolzenberger are going to look at some biomarker development in patients with metastatic kidney cancer. I think it's very important so that we can have some sort of way to tailor therapy and find out how to help those patients that do respond to respond better, and those patients that don't respond, why they don't, and what to switch them to. So I think those are important grants to, and we're very pleased to award those. So two of the other awards we're going to talk about today, and again, we talk just to reiterate $50,000 from Kidney Cancer Canada through your donations, which is very important to further kidney cancer research. You can always go on the Kidney Cancer website, and there's a tab there for earmarked for research, and we encourage you to donate to the research initiatives. And the 2 winners that we're going to announce, that were announced a couple of weeks ago, that we're going to have a talk from today, are from Dr. Keith Lawson at University of Toronto. He's going to talk about interrogating the single-cell molecular landscape of renal cell carcinoma metastases and the discovery of novel therapeutic targets. Now, Keith has been an amazing MD, PhD researcher. We're really, really proud of him. He's done a great amount of work in kidney cancer, and we did want to get him at McMaster, but he turned us down and went to the University of Toronto, but he's still up for grabs when he graduates, so maybe we'll get him back to Hamilton, or he'd be an asset wherever he goes in Canada. The other project that we wanted to just mention, who's not able to talk today, is from Kidney Cancer Canada, is Dr. Nida Da Costa of UBC, talking about her project is on identification and validation of comprehensive gene signature to determine treatment response and guide precision oncology in patients with metastatic clear cell carcinoma. And this is another project looking at how to tailor therapy for kidney cancer patients. And we do now know that there's a genetic component for kidney cancer. The genetic component is very big in prostate cancer and bladder cancer, and certainly kidney cancer is no slouch. Where there is a genetic component, we do know we have guidelines. Patients that have bilateral kidney tumors, tumors that are that have a family history, non-clear cell histology, and young patients under the age of 45, they should have mandatory genetic testing. So if you're in that group and you haven't had, talk to your doctor about getting genetic testing. They're available more widespread now. And her project is going to look at some of these gene signatures as well to tailor therapy for our patients. Now, I'm going to talk a little bit about Kristen McAlpine as well. Again, Kristen McAlpine graduated from the University of Ottawa. She was involved in our network as well, some great project looking at small renal masses, and now she's at back to the University of Toronto to look at some kidney cancer research and oncology research in her fellowship. And we're also going to get her back from McMaster someday as well. So looking forward to this talent coming to us someday. Before we get to their projects, and what they're going to talk about, I want to introduce Helene. Helene is going to speak briefly on her experience being on the grant selection committee. As you know, this is an objective committee where we have members from the network, where we have doctors such as medical oncologists, urologic oncologists, but we also have a partnership with patients as well to advocate for these projects that would benefit patients. They all benefit patients to some degree, but also to have a patient perspective on the grant panel. So Helene is going to talk to us a little bit about her experience with that.
Recently, I was asked to be on a panel for the SOPR 2020 Kidney Cancer Research Grant, not only as a board member of Kidney Cancer Canada, but to represent the interest of kidney cancer patients. A few years ago, I've learned about KCRNC, the Kidney Cancer Research Network of Canada, and their fantastic work with researchers across Canada on kidney cancer. Quite reassuring for a patient like me that some doctors, students, and researchers are working hand in hand to find a cure for my disease in Canada. I was excited to participate with this selection committee to give 250,000 grant from the SOPR family. We had 8 applications to study, read, and comprehend. It was a long process. I took this process very seriously, even if I did not have the knowledge and scientific background of the doctors and researchers on the panel. As I was reading these applications, all I had in my mind was the interest of us, the patients. How these projects would help us, how these projects would be good or not for us, how can we benefit. When the committee met, and I really felt that patients' opinion and point of view were considered in the decisions process. The 2 winners of the SOPR 2020 Kidney Cancer Grant were finally announced at the KCRNC Virtual Forum. It gives me hope that with all the research in progress and coming in, we are closer to find a cure for kidney cancer.
Wonderful. Thanks, Helene. That was wonderful. And again, we really appreciate your involvement in the grant deliberations. I should mention it was a long process, but again, an objective process. And I'm going to now turn it to Kristen McAlpine, who's going to talk about a patient decision aid for complex localized renal masses. Thanks, Kristen, and we're all really proud of all you've accomplished so far in your young career.
Thank you very much. Okay, just gonna bring my slides up. Okay, so thank you very much, Dr. Kapoor, for the introduction, and thank you very much for the committee for inviting me to present to you today. As has already been mentioned, my name is Kristen McAlpine. I'm a urologic oncology fellow at the University of Toronto. And today I'll be presenting a project that was completed while I was a urology resident at the University of Ottawa last year, entitled Shared Decision Making for Patients with a Small Renal Mass, Development of a Novel Patient Decision Aid. Before I begin, I would just like to say thank you very much to all of these agencies that have been there to fund our research, particularly to KCRNC, who provided me with a trainee research award while we were completing this project. We'll begin with the case of a 77-year-old female who is found to have a renal mass. She's healthy apart from some high blood pressure, active, and is retired. A CT scan shows a 2.5 centimeter mass in the posterior aspect of her kidney. She is referred to see a urologist and has many questions as the mass is causing her a significant amount of anxiety and she would like to have it treated. Patient decision aids are clinical tools that are used to facilitate shared decision-making for a specific population of patients who are facing a challenging decision. At a minimum, a decision aid must explicitly state the decision that needs to be made, offer the management options, including their outcomes, and help patients clarify and communicate their values and preferences. Decision aids have been created for many disease processes, and they have been shown to improve patients' knowledge, their accuracy of risk perception, their involvement in the decision-making process, and satisfaction with the care that they receive. Decision aids have also been shown to decrease patients' decisional conflict, their indecisiveness, and the variability in care that they receive. So why do we need to create a decision aid for patients with a renal mass? Well, in this study, which was a survey of patients and their caregivers who had previously undergone treatment for a kidney mass, 81% of responders felt that there were inadequate resources for kidney cancer patients. In fact, in Canada, kidney cancer experts and patient advocates found that the development of decision-making tools for patients with kidney masses was one of the top 10 priorities for kidney cancer research moving forward. As such, we sought to develop a patient decision aid to facilitate shared decision-making for patients with a small renal mass. To do so, we followed the Ottawa Decision Support Framework and the International Patient Decision Aid Standards, which set an evidence-based process to develop patient decision aids. To begin, we had to define the patient population that we wanted to include for the decision aid, and we felt that individuals with a small renal mass who were deciding between management options were the optimal patient population for this decision aid. Next, a team of urologists, patient advocates, and individuals who with experience in developing decision aids came together and decided that the following were the management options that should be included on this decision aid: active surveillance, partial nephrectomy, radical nephrectomy, and tissue ablation. As not every patient would be eligible for each of the management options, a page was included on the decision aid where a urologist could indicate to the patients which option was available to them. So for example, if a patient was eligible for an open partial nephrectomy, tissue ablation, and active surveillance, the urologist could indicate this to them, and then when they worked through the decision aid, they would know which outcomes to focus on. Next, we completed a thorough review of the literature to identify the best available evidence for outcomes of each of these treatment options. The following benefits were included on the decision aid: kidney cancer survival at 5 years, no evidence of kidney cancer at 5 years, and the length of hospital stay after each treatment. And the harms that were included were the rate of kidney cancer spreading or metastasis, the rate of major complication after treatment, the rate of urine leak and bleeding, the rate of dialysis, or the rate of flank bulge. After completing the review of the literature, the outcomes were synthesized on the decision aid using diagrams and figures. So for example, under radical nephrectomy, you can see that there are 100 faces for a given outcome, and the number of faces that are shaded indicates the percentage of patients that we would expect to have a given outcome. Patient advocates also indicated that they would want more information on the expected scars after each treatment option, and so we included this on the decision aid as well. Patients' values and preferences were clarified and communicated using this validated scoring tool, where a patient could indicate which outcomes were the most important to them and which were the least important to them when they were considering their management options. Once we completed a draft of the decision aid, we then moved forward with what is called acceptability testing. At this step in the development process, the individuals who will use the tool in the future, so patients and urologists, review the draft of the decision aid and give feedback on whether they feel it is acceptable for future patients. Patients and urologists completed a 10-question paper-based or online survey about the decision aid after reviewing it. The following slides will show the results of the survey that we performed. On the x-axis, or down at the bottom, you can see the percentage of patients who responded to each question. And on the y-axis are the responses. So as you can see, the language was felt to be easy to follow by the vast majority of responders. The length of the decision aid was felt to be appropriate. Most individuals felt that the decision aid provided a balanced approach to the different management options with regards to the amount of information, and that almost all individuals felt that this would be a useful tool for future patients referred with a small renal mass. Using the results of the alpha testing, we updated the draft of the decision aid and have completed a final version of the patient decision aid. This decision aid is available on the Ottawa Hospital Research Institute's website for free use internationally. It is also published in the CUAJ, the Canadian Urology Association Journal, in December of 2020, so next month. And recently was endorsed by Kidney Cancer Canada and placed on their website as a new resource for patients and caregivers. So an individual could click on this link and would be able to work through the decision aid, either on their own at home or with their urologist in the clinic. Going back to our patient, she was given the options of a minimally invasive partial nephrectomy, tissue ablation, or active surveillance. She had the opportunity to work through the decision aid and look at the various outcomes. She worked through the tool that allowed her to clarify and communicate her values and preferences, and decided to proceed with tissue ablation. So we have created a novel patient decision aid for patients with a small renal mass considering their management options. This tool is acceptable with patients and urologists and has been endorsed by Kidney Cancer Canada. And we plan in the future to convert this to an online tool where a patient could click through the outcomes. I'd like to say thank you to the entire team that was involved in creating this decision aid. And again, thank you very much for your time and your attention. And for these sponsors, if there's any questions, I'd be happy to answer them now, or please feel free to email me in the future as well. Thank you very much.
Wonderful, Kristen. Thank you so much. I'm just going to see if there's any questions on the chat here, so I can figure this out.
No, there are no questions on the chat.
Okay. All right, wonderful.
So, Anil, I have one comment. So, so as Christian mentioned, Kidney Cancer Canada has supported this and appreciates this valuable research that she's conducted. So, back in her Ottawa days, when we appreciate the work that Ottawa Hospital did with Luc Lavalier and Giselle, and we see that the exceptional value that this brings to our patient community. So Kidney Cancer Canada not only has the links on our website, but we will also be printing the decision aid in both English and French. So it'll soon be translated for all of Canada to be able to use and leverage with their diagnosis in determining what their future treatment perspectives could be. So thank you, Christian.
Thank you very much.
Wonderful, Kristen. Thank you so much. I want to acknowledge the full committee at this point. The chair was Dr. Christina Cannell, and again, Helene was on that. Michael Jewett was also a member of that. Andrew Weller, also to acknowledge his efforts reviewing these grants. And Fred Pouliot out of Quebec was also a grant reviewer. So thanks to all of those reviewers for reviewing, putting the time in to review these grants. So now we're going to have Dr. Lawson talk about his project. Keith, you have the podium.
Fantastic. Thanks so much. Can you guys see my screen?
Yes.
Fantastic. So it's an absolute pleasure to be able to present our work to you all today. I wanted to really start by thanking KCNRC as well as KCC for supporting our group over the years. There are a number of projects that we've been fortunate enough to partner with KCNRC for, and I think it's very important to keep basic science and discovery science kind of at the forefront of what we're doing in terms of research priorities. So I really do appreciate that, and we've been very successful at getting some initiatives off the ground based on these early funding initiatives. So we're very grateful for that. I'd also just like to take a moment to thank Dr. Tony Finelli, who's been a mentor and chair of a lot of the initiatives going on at the University of Toronto that I've been fortunate to be a part of, and also a recent chair he's been awarded that had been previously held by Dr. Michael Jewett, the Annalisa Farquharson Chair, which has also been very important for kickstarting a lot of these initiatives. So I'm going to be sharing with you a story today that really highlights one of the projects we're focused on, which is trying to identify new drug targets to prevent kidney cancer metastases. And I think I wanted to really start by first highlighting that there's a lot of encouraging work being done in the treatment of metastatic kidney cancer. As you know, patients and caregivers alike will know, there's a number of new options we have that are really improving patient outcomes with real benefits with respect to progression-free survival as well as overall survival. But I think at the end of the day, and it's been mentioned a couple of times now, what we really strive for in this disease is finding a cure for everyone. I think one of the most important ways of doing that is really going to be preventing kidney cancer spread or metastasis or progression to those later stages altogether. And historically, this has been done by the use of something called adjuvant therapy, and this is drug therapy that's given after surgery to prevent the spread of kidney cancer or the recurrence of kidney cancer. And unfortunately, most of the clinical trials, or I should say all of the clinical trials that have been done so far in this space, have not been very effective in improving overall survival for patients. So there's clinically really a need to find new adjuvant therapies or new therapies that prevent spread and prevent metastases. And our group and many others are very focused on trying to better understand the biology of kidney cancer metastasis, as we really think a better understanding of the basic mechanisms of how kidney cancer spread will shed light on new drug targets for this area. So taking a step back as way of background, it's important to recognize that in all cancers, not just kidney cancers, kidney cancer cells exist within the tumor with a number of normal cells. And I've listed a couple of different types of cell types here on this slide. This includes cell types from your immune system, fat cells, and other support cells. But these cells really exist in an ecosystem. And what's been very well recognized in the basic sciences is that in order for cancers to spread outside of their primary site, for example, jumping from the kidney to the liver, it really requires an interaction of cancer cells with these various normal cell types. And this is particularly true in kidney cancer, as studies have shown that tumors emanating from the kidney are really filled more so than other cancer types with a lot of these normal cells. So it's very important for our experimental approaches to really capture these dynamic interactions between cancer cells and normal cells. And this has really been one of the difficult areas of research because our traditional approaches, particularly in genetics, so looking at the genetic composition of tumors, have been unable to capture this dynamic crosstalk or communication between cells within the microenvironment that allow them to escape. So again, I'm just highlighting that this is one of the major barriers currently in our existing technologies. And the problem with this is really highlighted by an analogy that's really commonly used in the field, which is called the fruit salad analogy. And if we imagine a tumor is made up of different cell types analogous to this table or platter of fruit, all having a different diversity of fruit types, the problem with our traditional experimental or genomic approaches is when we sample the genetic information in this tumor, we actually get a blend of it. And in doing so, we mix all the different results from these cell types or fruits into a smoothie. And in doing so, we lose information that tells us what genetic signature is coming from what cell types. And if we don't know what genetic signature is coming from what cell types, we lose the ability to say and see how the cells are talking to one another. So this has recently been a problem that's been very nicely overcome by a new technology which is completely revolutionizing genetics called single-cell genomics. And what single-cell genomics does is it's able to isolate each individual cell within the microenvironment and couple that to a genetic read. And this really takes advantage of something called microfluidic technology. You can see this GIF GIF that I've pulled off YouTube, which really highlights nicely cells coming through this microfluidic device and being encapsulated by these liquid droplets. So these liquid droplets allow the capture of a single cell, and they actually have fluidic systems that breathe all the reagents to do the sequencing and genetic chemistry required for these experimental assays. And in doing so, it couples the individual cell to the genetic readout. So in doing so, we no longer get a smoothie blend of the genetic information. We really get a crisp fruit salad view of what's going on in the tumor microenvironment. And again, this is very important for being able to capture that crosstalk between cells. So this is giving us completely new genetic information from tumors that we've never been able to see before. So it's an incredibly exciting technology that everyone in the world is really getting behind right now. And of course, this is now, being used to study kidney cancer as well. And the nice thing about the study that we're proposing is we want to use and leverage this very powerful technology to study kidney cancer metastasis. And at the UHN, we have a very unique cohort of tumors that we've been collecting over the last 20 odd years or so, where we've taken patients' tumors from the time of surgery, from their primary tumor, as well as patients that have had surgery on their metastases, and stored them in a biobank. And we've stored them in a way that we can use single-cell genomic technology to start interrogating the genetics of how cancer cells can move in cooperation with normal cells from primary tumors to the metastases. So we're using this technology now to explore the genetic landscape at single-cell resolution in the primary tumors as well as metastases. And importantly, we have a cohort of tumors that have not metastasized. So we know, and we've followed these patients' tumors for a very long time, and we know they have not metastasized. So by applying single-cell genomics to these controlled tumors as well, we really hope to identify new genetic signatures of metastasis. And what I mean by genetic signatures, we really hope to identify genes that might be actually driving or causing metastasis. And that's the primary goal of the study that was recently funded by the KCNRC. And I want to really end by highlighting the ultimate goal of this type of work is really to identify biomarkers. Would be number one up front, but in the long term would be actual therapeutic targets that could be leveraged and made into drugs that could be used to prevent metastasis altogether. And this is a multi-step process, and I highlight this slide at the top by being bedside to bench and back, and I'm going to walk you through what I mean by that. So the project that was funded was really the first step. We need to use direct surgical samples, patient tumors, and breathe the best genetic technology we have into those samples to discover genes that may be associated with metastasis. And this is what the project that's funded is all about. We're trying to come up with genes that have never been really discovered before that could be driving this biological process. But it's important to remember that in order to then take that into a drug discovery pipeline, there's a number of other important steps. The next step after that would actually be taking it back into the lab and modeling what we see coming out of patient tumors. And this is very important to develop models that reflect what we see in the patient's tumor, such that we can actually go down a series of experiments to validate those genes as being proper drug targets. And I'm actually very fortunate to say that we have another Kidney Cancer Research Network of Canada funded project that's really aimed, all focused on trying to validate and improve the models of kidney cancer tumors. So we have a project now funded for the discovery phase. We have a project now funded for the modeling phase. And the last step would really be using new technologies like CRISPR, where we can actually go into the genetic code and selectively remove genes or turn genes on and see whether or not that actually disrupts the metastatic process. And there's a number of projects we have ongoing in the lab right now that really are focused on getting this technology going for kidney cancer. And I highlight this three-pronged approach as this is really an overall pipeline that we're developing in our group to be able to go from discovery genetic work on patient tumors to modeling that in the lab, and then ultimately validating some of these targets such that we can actually go down the process of making drugs against them. So we're very excited about this program. I think it's very important to remember that, you know, in order to come forward and make cures for kidney cancer, we need to really have this bench and bedside approach where we're kind of going back from the clinic to the lab and really working through all the deep biology that's involved in these various processes that are clinically important. And it's very encouraging that we've also been able to, with our funds, attract a lot of different multidisciplinary scientists. Their faces are up here on the map to kidney cancer. So, you know, initiatives like this are not just important for getting the individual projects going, but they're very important for team building and getting other scientists that have not historically been involved in kidney cancer focused on kidney cancer. So we're very fortunate and very thankful for all of your support, and I'm absolutely happy to take any questions. Thank you very much.
Well done, Keith. That's very impressive work, and we look forward to translating that to the clinical scenario. So I think that's where I'm going to go next. Is talk at the beginning of your. I know we have for Christine. I know we have until 12:45, and it's 12:50, but maybe just a few minutes for questions, and then we'll take a 5-minute break. So Keith, in terms of adjuvant therapy, right now we've had a lot of studies looking at patients who have high-risk cancer, patients who have their T3 cancers removed, where they have a 12-centimeter cancer removed, many maybe on the line as well. And then we've always talked to them about adjuvant trials, that many of them on the line may have been involved in some of the adjuvant trials, which adjuvant was open, adjuvant sunitinib, adjuvant TKI therapy, all negative trials now. No real advantage to going on a drug after you have had your kidney cancer removed. We've had some adjuvant adjuvant immuno-oncology trials that are closed, and we'll see what adjuvant nivolumab, adjuvant pembrolizumab shows in 2022. What do you think about where your work is going to go to help us get to that point where we have a drug tailored to a patient who has a high-risk cancer that we have a high probability of coming back, and how we can help them prevent that from coming back?
Yeah, so that's a fantastic question, and truly that is the goal of this research project. I think what our aim is here is to really better understand the biology. And I think because there's been a lot of advances in basic sciences and genetics, we're starting to see that there was a lot of information that was previously not known to us in terms of potential targets. So this is really an exciting time for us to use these technologies to really uncover completely brand new biology. So that would be the hope here, is to really, work through discovery projects and find new targets for patients. And ultimately the goal would be to have a therapy that would impart a long-term survival advantage with respect to preventing recurrence, preventing metastasis. So I think the goals are very aligned there. And at this point in time, we're really trying to look to these new tools to point us in the direction to really give you a solid answer to that question. So I think it's a time of optimism, and there's a lot of good work going on. And hopefully within the coming years, I have a really good answer and a target for you. And that's the goal here.
Wonderful. So it sounds like you're asking for more money in the future as well. I'm getting prepared for that. Great work, Kristen. Great work. Well done. Mona, if there's no other questions, I think we'll close the session.
Yeah,
so you're on silent.
And thank you for joining us.
Yes.
Your work is very valuable. Oh.
A question for Keith is a question from Russell. Is your research a follow on the existing DNA and RNA genomic profiling, like 500 gene panel?
Yeah, so I think that's a really important question. So there's a lot of work going on in genetics and a lot of existing genetic studies that have been done, particularly even in kidney cancer. And one of the important ways of leveraging our existing project is actually to go back to some of those traditional genomic tests and layer them into our study. And that's exactly what we're doing. And that's an important thing because single cell genomics is still very, very expensive. So one way that we hope to eventually translate this in the form of biomarkers would be using our single cell genomic data set to really narrow down some of the more important genes that we can look at in combination with some of the traditional approaches like Russell's mentioning here, and roll that out on a larger scale. So I think there's a lot of complementary things to learn in terms of leveraging single cell genomics with the traditional genomics approaches, which are certainly still here to stay without question, I thought.
Thank you.
And I
One
have
more,
a
Mona.
question for me. Yeah, that's the only one after. What is the timeline of seeing the single cell genomics advances in the patient world?
Another great question. I'm hard pressed to give a time because I just don't have a crystal ball. One thing I will say is that I'm very optimistic that soon we're going to see real discoveries being made with this technology that could change things for patients. Just because if you look across the sciences, with single cell genomics, we're discovering new cell types altogether by looking at standard questions that have been looked at for decades and decades in science, and we're uncovering completely new things using this kind of technology. So it's a very powerful discovery tool, and our job is going to be to make sure it gets applied to the disease that we all care about, kidney cancer, and then ultimately translated. So that does take time, but I do, and I'm very hopeful and optimistic that there's going to be a lot of new discoveries made in the coming years that hopefully we can start to translate and go down that process.
Yeah, I think Keith is going to have a great career in politics, because I'm not sure what the answer to that question was, but it's not like there's a lot, hopefully not too far off.
Yep.
There's been great strides made since I've been doing the research projects with the KCRNC and Kidney Cancer Canada, and I can remember your first presentation, Keith, a few years ago, and it's amazing the work that you have been doing and how far you've come. So it's very, very encouraging for our kidney cancer patient community. Thank you so much. And thank you, Kristen, because you have offered also a very important part of for kidney cancer patients, the decision aid. They're tough decisions we have to make, and all the information that we have at hand to help us make those decisions is most valuable. So thank you so much for both of you.